Good evening, ladies and gentlemen. I'm Coach Spivey, and we're back again with another amazing science tutorial video. And once again, I'm excited because I get a chance and opportunity to reach and teach to our youth today so they can build that big, bright, positive future for us tomorrow. But before we get started with our video for today, I want to give a shout out to my whole 7 period. They've been really on it as of late. So shout out to Nikeria Albritton, Taranisha Brown, Shamaria Butts, Joshua Chapman, Danae Hands, Justin Harrell, Katora Haynes, Nadia Lucas, Antavius Middlebrooks, Jatavius Scott, Jaquino Solomon, Tanisha Sullivan, Mackenzie Thorpe, Xavier Threet, and Elena Young. These young kids, they really inspire me and help me do what I do. So let's go ahead and get started with our video for today. And today we're going to be talking about viruses. So let's begin. So let's take a look at the discovery of viruses. In 1897, Dutch scientist Martinus Bijgerink called tiny particles in the liquid extracted from a plant disease viruses, which is actually the Latin word for poison. In 1935, American biochemist Wendell Stanley isolated crystals of tobacco mosaic virus, and he concluded that living organisms do not crystallize. In other words, he was saying viruses are not truly alive. Now let's look at the chart to see if viruses are living or non-living. So here are our characteristics of life right here. Then here's our virus versus our cell. If we look at the structure of a virus, it has DNA or RNA which is copied. Some have an envelope. If you look at the structure of a cell, it has a cell membrane and cytoplasm. And eukaryotes also contain a nucleus and many other organelles to help with cell function. If we look at the reproduction of a virus, they can only produce within a host cell. So viruses cannot reproduce by themselves. They need a host cell in order to reproduce. A cell reproduction is independent cell division, either asexually or sexually. So cells can reproduce independently on their own. So they can do this asexually without a partner or sexually with a partner. And then let's look at the genetic code. Genetic code for viruses can be the DNA or RNA. The genetic code for cells is just DNA. And do viruses grow and develop? They do not grow and develop. Cells, yes, they do grow and develop in multicellular organisms. Cells increase in number and differentiate. So that means when they differentiate, they perform different functions. So the cells for your liver, your heart, your spleen, your spine, your bones, all those cells differentiate because they have different functions. And do viruses obtain and use energy? No, they do not. Do cells obtain energy? Yes, they do. Do viruses respond to the environment? No, they do not. But do cells respond to the environment? Yes, they do. So if there's too much water inside the cell, some of the water will leave. If there is not enough water in the cell, some of the water will actually enter the cell. So they respond to that environment. And then do viruses change over time? Yes, they actually change and mutate over time as they evolve with the host cell organisms that they're living inside of. And then our cells change and evolve over time as well. So viruses are not considered living, and some of the reasons why: a virus is not a bacteria, fungus, protist, plant, or animal. A virus is a non-living particle made of proteins, nucleic acids, and sometimes lipids. A virus can only reproduce by infecting living cells. So a virus cannot reproduce on its own. It needs living cells to infect to make copies of itself so it can reproduce. And then a virus uses the organelles and enzymes of the cells of the host to make copies of itself. So once it enters that host cell, it uses all of the materials inside the cell to make copies of itself to reproduce. So now it's time for your first check for understanding. And you're going to write a brief summary of why viruses are not considered living. You have a 3-sentence minimum, and you can go ahead, pause the video, and begin now. So now let's see how you do it in your first check for understanding. And in your first check, you could have talked about the following. You could have talked about the virus's reproduction, their growth and development, their ability or lack of ability to obtain and use energy, and their lack of ability to respond to the environment. So if you talked about any of the 4 of these or all 4 of these, then you are on the right track. A virus has 2 to 3 essential features. So viruses have a nucleic acid. They either have DNA, like this virus on the left, or they have RNA, like this virus on the right, but they do not have both. And they use this DNA and RNA to pass along their genetic information. And then they have a capsid, which is a protein coat surrounding the nucleic acid. And this capsid protects that nucleic acid or the genetic material, which we know as DNA. So here's this capsid right here, which protects their genetic material. And then some viruses also, in addition, have tail fibers, which are used to attach to host cells. So here are those tail fibers right here. And they use these in order to attach to the host cell so they can insert their viral DNA inside that host cell. Then some viruses may have a membrane-like structure outside that capsid called an envelope. So this is an extra layer of protection. So if you see this is a membranous envelope outside of that capsid, so which is an extra layer of protection. And some examples are influenza, chickenpox, herpes simplex virus, and the HIV virus as well. Then the viral shape of the virus is determined by either its capsid or its nucleic acid. So it's determined either by its capsid shape or the RNA or DNA that it contains. So two examples of shape are the icosahedron, which has 20 triangular faces. So the herpes simplex, chickenpox, and polio have this icosahedron. So if you look right here, they have these triangular shapes, those 20 triangular shapes on its outside, on its capsid. And then a helix is a spiral shape like DNA. So rabies, measles, and tobacco mosaic virus have that helix spiral shape. So if you take a look at this capsid right here, it's got that spiral shape to it. So now it's time for your second check for understanding. And you're going to label the three major structures of the following virus and tell the function of each. You have 2 minutes to complete the following, and you can go ahead and pause the video and begin now. Now let's see how you did on your second check for understanding. So if you notice, I have this nucleic acid in the middle. So one of the major structures of a virus is this nucleic acid, which can be DNA or RNA, and its function is to pass along the genetic material of that virus. And then secondly, we have the capsid. This capsid is this outer cover or this outer layer, and its function is to protect the genetic material, which is that DNA or that RNA for that virus. And then thirdly, we have the tail fibers, and their function are to attach to the host cell. So let's take a deeper look at viruses. And there are two types of viral infections. Viruses use their genetic information to make multiple copies of themselves inside living cells. Viruses that infect bacteria are called bacteriophages, which literally means bacteria eating. Some viruses enter cells and replicate, make copies of themselves immediately, and these are called lytic infection viruses. And then some viruses are initially in an inactive state, which means at that state they actually take their DNA and fuse it with their host cell's DNA to help make copies of themselves. But they kind of remain dormant and sit still within that host cell until they are triggered and become active. And these will be known as lysogenic viral infections. Let's look at the lytic infection cycle first. So first, a virus injects its DNA inside of a host cell and breaks down the DNA of the host cell. So here's that attachment of this virus to the host cell. It attaches and then it inserts its DNA inside of that host cell, and then it breaks the DNA of that host cell down. Secondly, viruses use the cell's organelles and other materials to make copies of itself. So if you look at this right here, it's using the organelles inside of its host cell and other materials and the genetic material to make copies of itself. So here it is happening right here. Synthesis of viral genomes and proteins, and then they're being assembled even further right here. And these are the different parts of that phage assembly or that virus assembly. So it has the head, the tails, and the tail fibers, which are being assembled inside of this host cell. Third, The newly made viruses cause the cell to burst, which is called cell lysis, and leave the host cell to infect other cells. So if you look right here, these viruses have been fully assembled. Once they're fully assembled, they leave the host cell, which causes that host cell to burst, which we said earlier, which is cell lysis. And then our examples of that are the common cold, influenza, rabies, and AIDS. Remember, in lytic infections, they enter that host cell, make copies of themselves, and once they make copies of themselves, they leave that cell almost immediately, kill the host cell, and move on to other cells. So let's look at how the lytic virus replicates. So first there's absorption and attachment to the host cell, and then there's injection or entry of the viral DNA. And then it replicates its viral parts and assembles them, and then it releases by cell lysis, which kills the cell. So let's look at it again. We have absorption, attachment. It injects its DNA or genetic material. Then there's replication of its viral parts, and they assemble. And then they release, which kills and is the death of that host cell. So now it's time for your third check for understanding. And you're going to write what is occurring at each stage of the lytic cycle in the diagram below. You have 3 minutes to do so, and you can go ahead and pause the video beginning now. So now let's see how you did on your third check for understanding. So first, the virus attaches to the host cell. Second, the virus injects its viral DNA into the host cell, and at this point it's actually breaking down that host cell's DNA. And then third, it replicates its viral parts, or makes copies of its viral parts using the host cell's genetic materials and its organelles. Fourth, it uses those genetic materials and those other organelles in order to assemble its viral parts into other viruses. And then fifth, it releases from those viruses from the cell, which kills the cell, which is known as cell lysis. And this is the lytic cycle. Now let's take a look at the lysogenic infection cycle. So first, a virus injects its DNA inside of a host cell where it attaches to and is copied with the host cell DNA without damaging the host cell. So let's take a look. Here's that virus right here. It attaches to that host cell and it injects its DNA into that host cell. And its DNA attaches to the host cell DNA without damaging the host cell. Second, the virus DNA may replicate with the host DNA for many generations or for many years or months, which infects all of the daughter cells of the original host cell. So let's take a look. Here's that viral DNA right here. As the host cell replicates and makes copies of itself, it's actually replicating and making copies of the DNA from the virus as well. So let's take a look. There's the viral DNA, and then the host cell is making copies of its DNA, which in turn is making copies of that viral DNA as well. Then third, environmental factors and certain chemical triggers the virus to become active. It removes itself from the host cell DNA and directs the synthesis of new virus particles. So if you look at this point, this is where the lytic cycle begins. So certain environmental factors could be radiation or heat, and then chemical factors could be certain types of food that can cause that dormant DNA or that sleep DNA to wake up, leave the host cell DNA, and then start production of making new viruses using the host cell's DNA and organelles. And then if you look right here, those viruses are being fully assembled. And then fourth, lysogenic infection turns into an active lytic infection and bursts from the host cell and infects other cells. So that's what you see right here. So now it's a lytic cycle going on right here. And then at the end, we have all of those viruses. They burst from their host cell. They kill their host cell. Remember, that process is called cell lysis. And then these viruses release and they attach to other cells, and then the process starts all over again. And our examples of lysogenic infections are HIV, herpes, and hepatitis B. So now let's take a look at the lysogenic cycle. So now here's our original absorption or attachment to the host cell. And then second, we have penetration or entry or insertion of this viral DNA into the host cell. Third, the virus attaches to the host cell DNA. So here's a viral DNA attaching to the host cell DNA. And then fourth, the virus replicates with the host cell DNA. So here's that replication of the virus. And then 5 factors cause virus to detach from host cell DNA and begin synthesizing new virus particles. So here are those new virus particles. And then 6, it released by lysis, which results in death of the host cell. So let's take a look at some advantages and limits or disadvantages of the lytic cycle and the lysogenic cycle. So the lytic cycle, an advantage of the lytic cycle is that replication of new viruses is fast. So the replication of making copies of new viruses happens very quickly. But however, the host is also immediately killed, preventing the viral genome from passing on to the next generation of host cells. So the viral genome doesn't get to make enough copies, as many copies of itself as it needs, and it kills the host cell in the process, so it needs to find another host cell so it can start the process all over again. And then if we look at the lysogenic cycle, many more viruses can be made because the viral genome is passed on to future generation of host cells. Remember, the lysogenic cycle doesn't kill its host cell immediately. It attaches its DNA to the host cell DNA, and then its DNA is replicated every time the host cell makes new copies of itself. So, but however, a limitation of the lysogenic cycle is that its replication takes longer because it is dependent on the host cell's replication. So it depends on the length of time that it takes for that host cell to replicate or make copies of its DNA. Now it's time for your 4th check for understanding. And you're going to write whether each statement describes a lytic cycle infection, a lysogenic cycle infection, or both. You have 2 minutes to complete the following, and you can go ahead and start and pause the video beginning now. Now let's see how you did on your 4th check for understanding. Number 1, this infection can stay in the body for years before becoming active. And that would be the lysogenic cycle infection. Remember, it can stay dormant or lay still in the body for years or months. So that's why it's lysogenic cycle. Number 2, this infection makes copies of its virus, bursts out the host cell, and invades other cells immediately. The key word is immediately, which lets us know that would be a lytic cycle viral infection. Number 3, examples of this virus are the common cold, rabies, and AIDS. So this would be the lytic virus cycle. Number 4, example of this virus are hepatitis B and HIV, and this would be the lysogenic cycle. And remember, HIV can stay dormant in someone's body for many years and then become active. Number 5, replicates copies of itself with the host cell DNA without immediately killing the host cell, and that's going to be that lysogenic cycle. Remember, the lysogenic viruses put their DNA and fuse their DNA with the host cell's DNA and make copies, and this can last for years. Number 6, injects its viral DNA, makes copies of its DNA to make more viruses, leaves the host cell and infects other cells. And this actually would be both, because both the lytic and lysogenic cycle viruses actually do the same thing. They inject their viral DNA, make copies of that DNA, and then that DNA leaves the host cell and infects other cells. Ladies and gentlemen, I hope this science tutorial video was helpful and beneficial to you. I'm Travis Spivey, signing off with my son Jordan Spivey. Peace, and remember to have a wonderful, awesome, positive day.